Welcome back, everybody. In this lecture, we'll be talking about the regulation of gene expression in eukaryotes. The genetic information of a human cell is far more superior than that of a prokaryotic cell. It may occur to be about thousand-fold greater than what you observe in a prokaryotic cell. Additionally, it is a complex system out there because of the variety of cells that can be found in an eukaryote, where each cell type must express a particular subset of genes at different points in an organism's development, thereby indicating a complexity in regulation and also malfunctions of which can also cause diseased conditions. There are three ways in which an eukaryotic gene expression can be regulated. Number one is alteration of gene content or position. Number two being transcriptional regulation. And number three, alternative RNA processing. So, all of this will occur at the level of DNA replication or at the level of transcription or at the level of post-transcriptional modification and also at the level of translation. So moving on to the first step, that is alteration of gene content and position. The copy number of a gene or its location on a chromosome can greatly affect its level of expression, thereby meaning that more the number of genes on a chromosome or more the copies of the same gene on a chromosome may indicate greater expression of that gene. And this gene content or location can be altered by gene amplification, diminution, or rearrangement. Getting to know gene amplification better, the expression of a particular gene can be augmented by amplifying its copy number, meaning that if you want to increase the expression of a gene, the cell can simply increase the number of copies of that gene on the chromosome. For example, histone proteins and ribosomal RNA are needed in large quantities by almost all eukaryotic cells. Therefore, the genes encoding histones and rRNA exist in a permanently amplified state. And such gene amplification can also present problems. And the best example for this is the use of chemotherapeutic drugs. Methotrexate is a standard drug for cancer and it inhibits dihydrofolate reductase enzyme, which is responsible for regeneration of folates to be used in the nucleotide synthesis. And in the case of tumor cells, they become resistant to this methotrexate drug because the gene encoding for dihydrofolate reductase is amplified or the copy number has been increased for this particular gene by several hundred folds, and this will result in more enzyme production or more dihydrofolate reductase production than what can be handled by the drug. Then we have gene diminution, where a gene whose expression is only needed at a particular developmental point or particular tissue may be shut off by gene diminution. So, whenever a cell wants a gene to be expressed only for a particular duration or only at a particular time point, the cell can always deactivate the gene once the job is done. The example for this is maturation of reticulocytes into the red blood corpuscles, where the reticulocytes are nucleated and they have a wide set of genes that are responsible for this maturation. However, these genes are not required in the RBCs. So, what happens? The genes are lost along with the nucleus of the RBC, which is degraded upon maturation. Then we have gene rearrangements, which are used to generate each of the genes encoding the millions of different antibodies that are produced by the B cells. This is a phenomena that is particular to the white blood corpuscles. And sometimes occurrence of a bad gene rearrangement may lead to improper gene regulation. And the best example for such improper gene regulation is a type of cancer known as Burkitt's lymphoma. It is a variety of blood cancer where the translocation of a segment from chromosome 8 to chromosomes that encode immunoglobulins will lead to the activation of a gene that transforms the healthy B lymphocytes into the Burkitt's lymphoma cells, or the B lymphocytes which do not have any regulation over their proliferation. Moving on to the transcriptional regulation, the regions of each of the different chromosomes are either packaged as a heterochromatin or a euchromatin. And as we have studied this earlier, heterochromatin is that DNA which is very tightly condensed and rendered inaccessible to the transcriptional machinery. Thereby, it would remain transcriptionally inactive. And the best example for such inactivation of a chromosome by heterochromatin formation is the formation of Barr body. In humans or in human females, the 23rd pair of chromosomes or the sex chromosomes are double X in nature. So, in order to avoid double expression of the X chromosome genes, one of the X chromosome will be inactivated or packaged into a Barr body by the means of heterochromatin formation. Next, we have the euchromatin, where the DNA is not condensed, thereby it is accessible to the transcriptional machinery. The regions of a chromosome that are maintained as hetero and euchromatin will vary depending on the cell type, and thereby enabling the cells of specialized tissues or specific tissues to express a particular subset of genes required for that specialized tissue function. Now, moving on to the transcriptional regulation through individual genes. Firstly, we have the transacting elements, which are proteins that participate in regulating gene expression, and they are often called transacting elements. At least 100 different proteins are known to be the transacting elements, where they play a general role in regulating the gene expression in a manner similar to the activation of lac-operon in the case of prokaryotes by the means of the catabolite activator protein and cyclic AMP complex. And these transacting factors generally have multiple domains that are required for activity, and these domains may also include DNA binding, transcription activating, and ligand binding sites. The DNA binding domains of the transacting elements recognize specific DNA sequences in a regulatory region of the gene. And these DNA binding domains generally consist of one of the three motifs. The first is known as the helix turn helix, the second is the zinc finger motif, or the third could be the leucine zipper motif. So, at this point, before I continue with the lecture, I would like to tell you that this particular part of systems biology coincides with protein structure and functions, where you study about protein functions and applications. So, I have planned to discuss this in detail so that you can apply the same for protein structure and function as well. So technically, I won't be teaching this in protein structure and function because I'm doing it in detail here. Now, coming back to the topic, these DNA binding proteins, which possess the motifs, bind with high affinity to their recognition sites on the DNA or with low affinity to other DNA by the means of hydrogen bonds or the van der Waals interactions occurring between the amino acid side chains and the functional groups in the major groove and the phosphate backbone of the DNA. The remaining portions of the protein are involved in proper positioning of the DNA binding domain and in making the protein protein contacts with other transcriptional proteins. So, getting into the details of helix turn helix motif, these are proteins with symmetric dimers that recognize a symmetric palindromic sequence on the DNA. And each monomer of this protein will contain a region in which two alpha helix are held at 90 degrees to each other by a turn of 4 amino acids. So, if you can see here in the diagram, the alpha helix is holding two cylindrical structures. One is black in color and the other is white, and these cylindrical structures are the monomers of the helix turn helix motif. And one set of the helix makes contact with about 5 base pairs in the major groove of the DNA, while the other set sits on top of the phosphate backbone and help in proper positioning of the helices to fit into the major groove. Next, we have the zinc finger motif, where the proteins possessing this motif may contain between 2 to 9 repeated domains that are centered on a tetrahedrally coordinated zinc ion. So, if you can see here, there is a zinc ion placed in the center, and across this, there are at least two domains that are located, and each zinc coordinated domain forms a loop containing a alpha helix, and this loop is called the zinc finger. There are two types of zinc fingers, the C2H2 finger and the CX finger. C2H2 finger basically comprises of three fingers that interact with the major groove of the DNA and wrap around it. Many transcription factors are found to contain this type of domain. Meanwhile, there are the CX fingers where the proteins with this motif bind as dimers to the major groove of the DNA. And many steroid receptors are found to have this kind of domain. Finally, we have the leucine zipper motif, where the proteins with this type of motif have an amphipathic alpha helix at their carboxyl terminus. On one side of the helix would be hydrophobic groups such as leucine, and they are repeated at every seventh position for several turns of the helix. Meanwhile, on the other face of the helix will be present charged and polar groups, which are generally hydrophilic in nature. They bind as dimers to the major groove of the DNA, and the two alpha helices of each arm enter the major groove and wrap around the double helix of the DNA. This is a common motif found in several oncogenes. And if you have any doubts, you can always contact me. Thank you.